Hello, my name is Hannah Jandrin, and I'm an undergraduate research assistant in Dr. Werner Geldenhuys's lab under the Department of Neuroscience at the WVU School of Medicine. My project focuses on analyzing MitoNEET protection against paraquat-induced oxidative stress in C. elegans aging models. MitoNEET is an outer mitochondrial membrane protein that is important for mitochondrial function. MitoNEET in previous research has shown promise to protect against oxidative stress. Oxidative stress occurs when there is an accumulation of reactive oxygen species, or ROS, a byproduct of ATP production in the mitochondria. Oxidative stress in previous findings has shown to be linked to neurodegeneration. There's little to no information, however, regarding MitoNEET's ability to protect against age-related oxidative stress. Understanding its protection in this model would allow a pathway to translate our findings over to age-related neurodegenerative diseases such as Alzheimer's disease. C. elegans, a species of transparent nematode worms, were used as an aging model for this study. Our goal was to understand the role of MitoNEET in age-related oxidative stress resistance and ROS production, specifically the most common ROS, superoxide free radicals. This will allow us to have a better grasp on how MitoNEET may have protective properties in age-related neurodegeneration. It was hypothesized that in knockout for MitoNEET populations, there would be a significantly higher amount of oxidative stress and ROS production than that of the wild-type control population. Additionally, it was expected to see a significantly lower amount of oxidative stress and ROS production in overexpressor populations than that of the wild-type control populations. These results would point toward MitoNEET as having protective properties in our aging model. To test this hypothesis, we used three C. elegans strains. Wild type is our control population, knockout for MitoNEET, and overexpressor for MitoNEET. Then the worms were aged into two distinct age populations, day 1 of adulthood and day 5 of adulthood, in order to compare how age may affect oxidative stress. The populations were then subjected to varying treatments: T equals 0 or basal oxidative stress conditions, 2-hour M9 buffer serving as our vehicle control group, and 2-hour 100 millimolar paraquat, which was used to induce oxidative stress. Paraquat is an herbicide known to induce oxidative stress. To assess oxidative stress, 3 assays were performed: Cellrox green staining, DHE, and MitoSox red. Cellrox green staining was used to observe overall oxidative stress. The more fluorescent the sample is, the greater oxidative stress that sample is under. In the day 1 populations pictured in figure 4a, increased fluorescence was observed in the paraquat-treated knockout group compared to the wild-type control, indicating MitoNEET is crucial to protection against paraquat-induced oxidative stress. In the day 5 populations pictured in figure 4b, overall increase in fluorescence across all knockout groups compared to controls indicates that increase in age may have played a role in decreased protection from oxidative stress. Also indicated by fluorescence, the paraquat-treated knockout group showed reduced protection, while the paraquat-treated overexpressor group showed increased protection from oxidative stress compared to the wild type, further indication MitoNEET protects against age-related oxidative stress. DHE assays were used to quantify superoxide production via relative fluorescence. Day 1 population staining showed a reduced relative fluorescence for overexpression of MitoNEET in comparison to wild type under paraquat-induced oxidative stress. Day 5 populations indicated that loss of MitoNEET increased basal and paraquat-induced superoxide formation compared to the wild type control, further indicating MitoNEET has protective properties against superoxide formation in an aging model. MitoSox red assays were used to quantify mitochondrial superoxide production specifically. Day 1 population staining showed MitoNEET overexpression reduces basal mitochondrial superoxide formation compared to the wild-type control. Day 5 populations showed that knockout populations for MitoNEET had increased basal mitochondrial superoxide formation, while paraquat-induced production is trending toward protection in the overexpressors. We are able to conclude that the loss of MitoNEET showed an increase in basal superoxide production in aged populations while also increasing mitochondrial superoxide in both young and aged populations. Overexpression of MitoNEET protected against basal mitochondrial oxidative stress in aged populations while also protecting against overall mitochondrial superoxide formation in both young and aged populations. For further directions in this research, we wish to analyze the loss of MitoNEET in neurodegeneration post-stroke and in Alzheimer's and Parkinson's diseases, as well as develop drugs that may protect MitoNEET integrity and increase MitoNEET. Thank you for taking time to listen to my project presentation. If you have any additional questions, please feel free to reach out to one of the emails at the bottom of this poster.